Bismillahirrahmanirrahim. In the name of Allah, the most beneficent, the most merciful. Assalamu alaikum everyone. Good morning and welcome to my presentation. First of all, let me thank you all for being a part of this meeting. Before starting, I would like to introduce myself. My name is Farzeen Mansha and the subject of my presentation is transcription. I'll take your precious 15 minutes to share my knowledge. And one thing, I would like to leave questions at the end of my talk. There will be time for Q&A session at the end. So, as you people know, the secret of getting ahead is getting started. So, without wasting time, let's get started. So before proceeding, I want to share with you guys the contents of this presentation. So first of all, I'll talk about cell division, then I'll discuss with you central dogma, then I build a mind map for easy understanding of transcription. After that, I'll share with you the etymology, discovery and definition of transcription. After that, I'll share you the salient features of transcription, then its requisites means what is needed for transcription, and at last I'll discuss with you its steps. So let's begin. So my dear class fellows, as you know, the topic of my presentation is transcription, but you may wonder why cell division is displayed on your screens. So let me show the relationship between cell division and transcription. So let's start from its definition. What does it say? It says the process in which parent cell divides into two or more daughter cells is called cell division. And for this thing, there are two mechanisms. One is mitosis and the second one is meiosis. In mitosis, same number of chromosomes is retained, while in meiosis, chromosome number is reduced to half. How all this gonna happen? I'm not concerned with this. I'm just relating cell division to transcription. So as cell divides, new cells generate. If number of cells is increasing, then there should be some mechanism for like transfer of genetic material and for its conversion into some other useful product. So the process which does all this is called central dogma. Definition, what does it say? It says information flows from DNA to RNA to proteins, and it is divided into two processes. One is flow of information from DNA to RNA, that is transcription, and the other is translation, that is flow of information to synthesize proteins. So all this scenario falls under the heading of central dogma. Now let's see its grammatical representation. So first one is DNA, that is double stranded, and it is converted into single stranded RNA through a process that is called transcription. And then this nascent single stranded RNA is further converting into amino acid through translation, and these amino acids are assembling to form a polymer that is called protein. So the whole scenario in which double stranded DNA forms a single stranded RNA through transcription, and then this single stranded RNA forms a amino acids which further assemble together to form a polymer that is called protein, and this whole scenario falls under the category of central dogma. Now it's time for building a mind map for easy understanding of transcription. So first point is synthesis of protein require DNA and RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. As we all know, DNA controls the life and activities of the cell, so obviously information or for synthesis of anything, like here we are talking about protein synthesis, requires DNA. And the second point, every protein has got unique amino acid sequence. When amino acid sequence is unique, then obviously protein have to be specific. Information about amino acid is in DNA. The information present is DNA is in coded form. DNA contains a number of genes. What is a gene? A gene is a unit of heredity. A gene carries specific base sequence encoding a protein. When we talked earlier that DNA controls the life and activities of the cell, and it is composed of genes. So obviously when proteins are specific, amino acids are specific, then obviously genes have specific base sequence for encoding that specific protein. And the next thing, three consecutive bases in gene codes codon, The codons are code for amino acids. Like for example, the three bases like adenine, thymine, guanine. If they are coming consecutively, then they form a group that is called codon, and this codon is a code for amino acids. So 20 amino acids have 64 codons. As we all know, there are 170 types of amino acids. Among them, 25 are constituents of protein, and among them 20 amino acids are of great importance, and these 20 amino acids codes for 64 codons. The gene is the code for amino acid. Information in genes synthesize proteins. Different RNAs synthesize proteins. So that was the mind map for easy understanding of transcription. Now move towards the main topic, that is transcription. So etymologically, the word transcript means copy, print or rewrite something. So we can say that it is the process in which messenger RNA copy of DNA is formed. So you may wonder what is like a messenger RNA copy of DNA. Suppose we have a document containing some information and we want to copy it, or we want to get its print. Similarly, DNA contains the information for life and activities of the cell, and we copy it in the form of messenger RNA. So that's the whole scenario behind this. So now discuss its definition. What it says: the synthesis of RNA molecules using DNA strand as the template so that the genetic information can be transferred from DNA to RNA is called transcription. And who discovered this? This process was elucidated by Roger Kornberg. Now it's time for discussing salient features of transcription. So first point is uses DNA as a template. You may wonder why DNA as template, because it carries the genetic information. That's why DNA is being used here like a template. And the second thing, phosphodiester bond is formed, a type of bonding that is formed in nucleotides. And the third thing, synthesis direction is from 5 prime to 3 prime. As we all know, DNA is double stranded, one, and these are antiparallel to each other. One strand is from 5 prime to 3 prime and the other one is from 3 prime to 5 prime. So this point says that transcription proceeds from 5 prime to 3 prime. Next point, no primer is required. So basically, first of all, we have to know what is primer. A primer is something that initiates a process. As we know, RNA polymerase is the enzyme used in transcription that can itself initiate the process of transcription. So it doesn't need a primer to initiate it. So there is no need of primer. And the next thing, in prokaryotes, it takes place in cytoplasm, and in eukaryotes in nucleus. So as we know that in prokaryotes there is no nucleus, only DNA is present that is submerged in cytoplasm. So obviously the process of transcription takes place in cytoplasm where enzymes are present, and the process of transcription takes place simultaneously with translation. Whereas in eukaryotes, the process of transcription takes place in nucleus, because RNA is synthesized in nucleus, which is further transferred to ribosome in cytoplasm for synthesis of proteins. So that's why transcription takes place inside cytoplasm in prokaryotes and inside nucleus in eukaryotes. And the last thing, only a short segment of DNA is transcribed. So you may question why only a short segment of DNA is transcribed, why not the whole genome, and why only its one strand is transcribed. So the students, and the answer is very simple, that only a segment of DNA is transcribed, and it totally depends upon the physiological need, development and environmental changes. That's why whole genome is not transcribed, and that region which is transcribed is called structural gene. And the next question, why only one strand is transcribed? So it seems reasonable that only one strand is used, because transcription of RNA from both strands would produce two complementary RNA strands from the same strand of DNA, and presumably it would produce two different kinds of proteins. So that's why only one strand of DNA is transcribed into messenger RNA. And now it's time to discuss its requisites, means what is needed for transcription. So the first thing is DNA, that is used as a template. So what is a template, and let's discuss this. The template strand is the strand from which the RNA is actually transcribed. And it is also called antisense strand. And the second thing, substrate like ATP, GTP, CTP, and UTP. We have talked about nitrogenous bases into two groups. One is purines that are doubling like adenine and guanine, and the second one is pyrimides which are single ring like cytosine and uracil as in RNA. And the last thing, enzyme, RNA polymerase. So as we all know that the word ASE is used at the end of an enzyme, so definitely it's an enzyme. And metabolism is like the sum of anabolism and catabolism. So and this is done with the help of enzymes. So the enzyme which does the anabolism of RNA, like the synthesis of RNA, is called RNA polymerase. There are three steps involved in transcription: initiation, elongation, and last one is termination. In initiation, RNA polymerase recognizes the promoter site and then starts the transcription. In elongation, RNA strand is continuously growing, whereas in termination, RNA polymerase stops synthesis and nascent RNA is separated from the DNA template, as you can see this in the figure. As I have told you about RNA polymerase in the previous slides, now I want to tell you that it comprises two parts. One is sigma factor that recognizes the promoter site, and the second one is coenzyme which is attached to template strand with the help of sigma factor. As soon as transcription starts, sigma factor is released and the rest of the function is performed by coenzyme. Like you can see, this is strand of DNA that is called template strand. It is from 5 prime to 3 prime, and RNA polymerase is attached to promoter site through sigma factor, and this is termination point. After attachment, sigma factor releases and the rest of the function is performed by coenzyme. There are two promoter sites for transcription like TTGACA, TATAAT, and promoter sites on prokaryotes and eukaryotes vary. The number is actually tell us about the position of nucleotides, and the sign of negative tells how many nucleotides or how many numbers this promoter site is away from the transcriptional unit. So that vary in prokaryotes as well as in eukaryotes. Prokaryotes have only one type of RNA polymerase. Eukaryotes have three types of RNA polymerase: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3, with synthesized ribosomal RNA, messenger RNA, and transfer RNA. So you can see in this picture the bulging portion is transcriptional unit, and this is like promoter sites. Two promoter sites, transcription starts from here, and we count it like 1, 2, 3, and this is at the 10th, and we may count this 10, 11, 12, 13, but we named this whole group as 10th element and this one as minus 35th element. So that was the diagrammatic representation of whatever I said in the previous slide. Elongation. RNA polymerase starts joining complementary ribose nucleotides to the 3 prime of DNA strand. As we talked, transcription proceeds from 5 prime to 3 prime. The same complementary bases paired in DNA, but in RNA, uracil replaces thymine, thus uracil complement to adenine. So in elongation, only RNA strand is continuously growing. Whereas in termination, transcription continues. Finally, RNA polymerase reaches the termination sequence, thus transcription stops. As you can see here, there is some stop codon for terminating this process. So as we know that nucleic acid is broken by nucleases into nucleotides, and phosphodiester bond is being broken by phosphatases into phosphates. So to prevent the breakage of this strand, there must be something. So what is done? A cap 7-methyl GTP is added to one of its side, and a tail named as poly A tail is added to its other side. So this changed the whole structure, and that's why now we can say it is protected. But now you may question that how RNA is separated from DNA. So for this there are some stop codons, like many simultaneous bonds between adenine and uracil that makes the attachment of RNA weaker, firstly producing a hairpin like structure as you can see in this figure, and then it permanently detaches its RNA, and RNA can now move to ribosome for synthesis of proteins. So here transcription complete. So that's my presentation. I try to make this concept easiest. If you have any question you can ask right now, and thank you so much for your interest, and Allah Hafiz.